I, Janusz Pawlishin, together with my co-workers and collaborators, are happy to introduce you to the advances in solid phase microextraction. Before we proceed, I would like to make sure that you understand the reasons why microextraction frequently matches sensitivity of exhaustive extraction techniques, where much higher volume of the extraction phase is used. There are two principal reasons for that effect. One is that the enrichment factor in microextraction is much higher, as we are not depleting the analytes from the matrix, as it is in the case of exhaustive extractions, where most of the analytes are removed to the extraction phase. And the second reason is that all the analytes extracted are transferred to analytical instruments. So at the end, the sensitivity is the same. This means the amount of analytes transferred to analytical instruments is identical. The other interesting advantage of solid phase microextraction is that it is a platform technology. This means you can coat with the extraction phase different surfaces. Here you can see the fiber, tubing, vessels, particles, steering mechanism, disc, thin films, and so on. That leads to different formats of extractions according to the applications, and leading to many different applications, like recently done on-site sampling, direct coupling to mass spectrometry, medical applications, and on-site analysis, even underwater applications. One of the most interesting developments is direct extraction from the complex matrices. It is already known that headspace SPME is very clean technique, because the analytes, in order to reach the fiber, needs to be volatile enough to migrate via the headspace, and therefore they could be easily transferred to analytical instruments such as gas chromatograph and analyzed without introducing any contaminants. There's opportunity to do the same in direct extractions, since most of the samples we analyze are aqueous-based matrices. So analytes can be extracted via free form, and as long as we eliminate the effect of the matrix onto the coating of the fiber, we can accomplish the fact that we have a very clean extraction, because all the components which can migrate to aqueous phase can be also very effectively transferred to a C or directly to mass spectrometer. This is accomplished by using protection layer over the sorbent. The protection layer is the biocompatible polymer which prevents adsorption of macromolecules in pieces of tissue onto the coating. There is additional advantage of using matrix compatible coatings in extraction of complex matrices, that we obtain so-called balance coverage, which has been discussed in more details in a paper listed below. But in summary, the objective of this is that when we place the fiber in contact with the sample, we collect both polar and non-polar analytes in about the same amounts, as polar analytes are present in the high concentration in the free form, as they are not bound to the matrix, while hydrophobic analytes are bound heavily to the matrix, resulting in the low free concentrations and therefore slow accumulation onto the fiber. In addition, as this slide shows, around the fiber we have a bonder layer, which provides additional protection against macromolecules reaching the coating.
Hello everybody, I'm Varun Singh, and in this section I'm going to discuss about coatings which are generally used in solid phase microextraction. In SPME, coatings are most important part during extractions. Coatings are responsible for extracting different analytes from diverse matrices. Nowadays, many commercial SPME fibers with different coatings are available. These fibers are generally made by coating these phases over silica fibers. An SPME fiber generally consists of a support, coating, and overcoating. In most of commercial coatings, support used is silica, but researchers have used quartz, nichrome, gold, silver, and platinum wires also. But these are expensive, fragile, and hard to modify sometimes. To overcome these problems, fibers were developed on stainless steel or nitinol fibers. These fibers are stable at high temperature, flexible, and can be easily modified. Commercial coatings, besides having advantages, had some limitations. To overcome these, new materials were utilized to make fibers for SPME. In the next slide, we can see the materials used in recent years. Researchers have developed fibers with carbon nanotubes, boron nitride sheets, covalent organic frameworks, graphene, graphitic carbon nitrides, metal organic framework, molecular imprinted polymers, and ionic liquid. Whereas carbon nanotubes and graphene are generally used for extraction of hydrophobic analytes in their primitive forms, but they can also be used in their composite form for extraction of polar and non-polar analytes from various matrices like water, environmental water sample, oil samples, food, etc. Whereas boron nitride, graphitic carbon nitride, covalent organic framework, and metal organic framework are class of compounds which come from porous materials, which provide ample of interactions like hydrogen bonding, van der Waals interaction, hydrophobic interaction, pi-pi stacking, coordinate bonding, acid and base interactions. These interactions provide unique property along with their unique structure to develop tunable coatings. These coatings can be applied in headspace or direct immersion mode to extract analytes from complex matrices. Whereas in order to perform selective extraction, researchers have focused on molecular imprinted polymers as the coatings, and reports on these have been discussed in this review. For extraction of ionic compounds and better enrichment, ionic liquids were used. They provide all the interactions like other materials in addition to ionic interaction, which can make them ideal candidate for coatings. In next slide, some selected application on these materials will be discussed. First application in this case is use of carbon nanotubes as a coating in solid phase microextraction on stainless steel fiber. These fibers were made in the case is a composite of ionic liquid and carbon nanotube. First, stainless steel fiber is coated by support material which can anchor carbon nanotubes on its surface. After that, ionic liquids were immobilized on the surface by pi-pi stacking. In another application, graphene oxide was immobilized over stainless steel fiber by chemical modification. These graphene oxide sheets were modified by hydrophilic and hydrophobic groups to perform normal and reverse phase SPME. By this way, we can use hydrophobic support and high surface area of graphene oxide or carbon nanotubes to perform extractions either from hydrophobic or hydrophilic matrices for extraction of hydrophobic and hydrophilic compounds. In this slide, we will see use of ionic liquid, metal organic framework, and aptamers as coatings in SPME. In first application, an hydrophobic ionic liquid was immobilized over chemically modified stainless steel wire by inducing reaction of vinyl groups of ionic liquid and thiol groups of dioctane thiol. Whereas in another application, polymeric ionic liquids were used for the extraction of mRNA. Metal organic frameworks made by using copper ions and benzene tricarboxylic acid were used in composite form with graphene oxide to form SPME fiber over silica substrate. In order to perform selective extractions, researchers also developed aptamer-based SPME fibers. In this case, aptamers were immobilized by using polyacrylonitrile co-malic acid modified fiber and were used for extraction of targeted analytes.
Hi, this is Ezel Boyacı from Middle East Technical University, and I'm going to summarize to you the recent developments in the SPME geometries. I will keep my focus on geometries that they have gained significant attention lately. Although the fiber geometry of SPME was the first introduced one, there is still important progress ongoing there. In this regard, there are three main focuses. The first one is improving the robustness of the fiber, and in this context, Superco has launched GCM-enable Stable Flex SPME fibers as an improvement to the fiber endurance by coating a flexible fused silica core with the same extraction phases used in the traditional fibers, which is also partially bonded to the core. More recently, Superco has launched SPME fibers based on flexible metal alloy that bring the stability of the SPME fiber to a superior level. In addition, CTC has launched the SPME Arrow, where a thicker extraction phase is coated on a relatively wider stainless steel rod, while the tip of the fiber is equipped with an arrow tip to easily pierce a GC septum. This modification increases the volume of the extraction phase, which eventually provides enhanced sensitivity and extra stability. And lately, recessed SPME fibers were introduced to the same regard. The main benefits of this fiber include the use of the well-protected thin extraction phase, enabling fast equilibrium extractions. The recession protects the extraction phase from possible damages as it penetrates through outer protective tissue such as fish scale, which makes it suitable particularly for in vivo applications. The second one is further miniaturization of the fiber geometry. Two approaches are critical here. The first one, miniaturization of the coating and the support to fit a single cell to capture its elusive information with tip diameter about 10 microns. And the second one is miniaturization of the coating to perform non-depletive extractions from a small sample volume such as 100 microliters. This is crucial particularly for pharma industry where the same sample can be monitored for pharmacokinetic changes. The third area is the development of new extractive phases suitable for thermal and solvent disruptions, with an ultimate goal of integrating two important analytical platforms, which are gas and liquid chromatography. With this aim, recently Teflon HLV-coated fibers were introduced, which have shown promising results for metabolomic and multi-residue analysis. The second area of development is in thin film SPME, which actually improves sensitivity of SPME by using larger extraction phase in a thin layer geometry, therefore improves the sensitivity without sacrificing the extraction time. So this geometry is further divided as thermal stable membranes compatible with the GC desorption using spatial desorption unit and solvent stable thin film blades compatible with LC. The most important development here is the coupling of the blades with mass spectrometry for rapid analysis and development of stable and thin extractive phases and their applications. Dispersed magnetic SPME is another geometry that gains a lot of attention lately. One of the most important benefits here is the use of nanoparticles, which can be dispersed fully in the sample in absence of magnetic field. Therefore, we can get the full benefit of particle surfaces as well as very fast extractions, which is based on radial diffusion. Also, magnetic feature of the particles allows the user to manipulate the particles with an external magnet. Here, the most important progress is in the development of the selective extractive phases for functionalization of the particles for specific needs. Stir bar sortive extraction has similar idea to the thin film microextraction to improve the sensitivity of the extraction, but as can be seen from the right figure, the extraction with this method can be longer compared to the thin film microextraction since this approach uses a thicker extractive phase. As can be seen, stir bars can be used both for stirring the sample and extraction of the nanolight simultaneously. The direct mode can provide extraction of semi-volatiles and non-volatiles, while the stir bar in the head space can be used for volatiles. Even two stir bars can be incorporated in the same sample to get the maximum information from the studied system. And as can be predicted, the most important progress in this geometry is the development of coating methods to improve the stability of the extractive phase to provide extra endurance against the friction between the container and the extractive phase. The other important developments are in preparation of new selective extraction phases.
Hello, my name is Emanuela Gianfrido, and I will discuss the section of this presentation dedicated to the use of SPME for food analysis. Historically, most of the application of SPME for analysis of food commodities were confined to headspace analysis. This sampling mode prevents the direct exposure of the SPME coating into complex food matrices, thus protecting the device from any unwanted attachment of matrix components that can affect its extraction efficiency. However, even if headspace SPME works quite well for analysis of volatile compounds, some limitation can be encountered when analytes with a limit vapor pressure, such as pesticide, should be determined at trace levels. In these cases, direct immersion SPME represents the best link sensing mode because it provides enhanced extraction efficiency. However, conventional coatings suffer precocious deterioration that leads to poor extraction performances when directly exposed to complex matrices. To overcome this limitation, many efforts were made towards the development of robust coatings for direct immersion in food samples that we define as matrix-compatible coatings. The matrix-compatible coating discussed in this section are constituted of a conventional divinylbenzene polydimethylsiloxane fiber overcoated with a very thin and smooth layer of pure PDMS that guarantees anti-fouling properties for the SPME coating. The improvement obtained by these matrix-compatible coatings over conventional SPME fiber is significant. Please focus your attention on the left part of the slide and notice the difference in morphology of a DVB PDMS coating used only 20 times for extraction in grape juice and a matrix-compatible coating used 120 times for extraction of grape pulp. In the first case, it is easy to notice the deterioration of the coating surface due to carbonization of matrix constituents. For the matrix-compatible coating instead, no noticeable changes in the coating morphologies can be noticed after 120 extraction. Moreover, any inevitable deposition of matrix component on the surface of a matrix-compatible coating is reversible. And this is very important because it implies that the coating can be simply cleaned with solvents of any lint-free tissue and be brought back to the original state. In order to further enhance the lifetime of these matrix-compatible coatings, two cleaning steps can be included in the conventional SPME routine: a quick pre-desorption rinsing step and a more thorough cleaning step after desorption that we defined as washing. This approach was successfully tested with a variety of food samples. What we could notice was that for matrix with high water and sugar content, a rinsing in pure water and washing a water-methanol mixture was efficient to ensure good cleaning of the coating. However, for samples with higher fat contents, such as avocado and soy milk, this approach was not really efficient. After an attentive optimization of the cleaning procedures, we found out that adding a small amount of organic solvent to the rinsing solution works very well to clean oily residues from the coating surface prior desorption, thus avoiding also any instrumental contamination.
Good day, everyone. So my name is Jonathan Grandy, and I'll actually be the person discussing our environmental analysis approaches with various SPME-based samplers. So the first application we're going to be discussing today is our on-site experiment performed with the portable GCMS for the free versus total determination of formaldehyde in car exhaust. With this, we were using one of the standard analyte generating vials that was preloaded with pentafluorophenylhydrazine derivatization reagent, particularly good for derivatizing carbonyl compounds such as formaldehyde. Now, in this experiment, we performed SPME extractions for determining free analyte concentration in car exhaust and needle trap extractions for determining the total analyte concentration. And of course, we could subtract the two to actually see the particulate binding of formaldehyde. So as you can see in the table in the bottom left, when the catalytic converter was still cold, we were extracting about 10 times the amount of formaldehyde than when the catalytic converter is heated up and functioning properly. This is unsurprising. It's what the catalytic converter does. However, what was a bit more surprising is we saw a reasonable discrepancy between the total amount of formaldehyde shown by needle trap at 0.37 nanograms per milliliter, then the 0.18 nanograms per milliliter seen with the SPME fiber, meaning we had about 51% particle binding. Originally, we thought this was a bit odd. However, once we considered that the particulate matter coming out of car exhaust isn't dry carbonaceous material, instead it's actually aerosol particles, we were very quick to realize that formaldehyde would have easily dissolved in this aqueous aerosol particles, giving us this free versus total differentiation. Moving along to solid sample analysis, I'm going to quickly discuss an application for the determination of PAHs from a certified soil sample. So for performing this, we were actually using our pressure balanced cold fiber SPME approach versus regular SPME. The reason for this is what we'd seen is in order to quantitatively liberate our analyte from the soil sample, we need to apply very high temperatures, such as 200 degrees Celsius. However, when we do this to a sealed vial, of course, that generates pressure inside of the vial, such that when we puncture that needle, we can actually lose our analytes and get very high RSDs. So to get around this, the pressure balancing, essentially, we're using a gas-tight syringe to first remove a predetermined amount of air from that vial prior to heating, such that once we reach that temperature, we have a good balance between the atmospheric pressure outside and the pressure inside of the vial. Actually, at 200 degrees Celsius, you might be able to see that by removing 7.5 milliliters from that vial, we hit a good balance at about 100 kilopascals. You can see that in the figure on the right. As you can see in the bottom left, however, when we are using our pressure-balanced cold fiber SPME approach, we saw our results were much more agreeable to the certified value from that sample, and RSDs were also much lower. In terms of water analysis, something that's been gaining quite a bit of traction is the use of high surface area thin film microextraction devices in lieu of a standard SPME fiber. So we've actually developed morphologies of these devices for both GC applications shown on the left and HPLC applications shown on the right. As you can see, for the GC applications, when we're actually looking for pesticides spiked into surface water samples, we can see a major improvement in sensitivity when we use the thin film microextraction approach versus US EPA certified liquid extraction methodologies and also our SPME fibers. Furthermore, when we actually compare the accuracy of these certified liquid extraction certified methods, we see comparable accuracy when we use our thin film membrane approaches. When we look into more HPLC amenable analytes, such as the benzophenone UV blocking agents that we're demonstrating in this study on the right here, what we were actually able to do is compare a TWA TFME device versus an open bed TFME device shown in the top right-hand corner, and then compare our instantaneous open bed TFME detections to the average over actually a 3-month period, using the retracted TFME TWA device. Now, of course, we don't get quite the sensitivity on that retracted device. However, when we are at those higher PPT levels, you can see that actually the concentration determined using the TFME TWA device sat about in the center of the values using the instantaneous open bed sampling. Furthermore, if we actually look at our GC amenable thin film microextraction devices, we've actually been able to couple these to our portable GCMS instrument, thus making up for the inherent loss of sensitivity we see when we miniaturize such a detector with a much more sensitive sampling approach. Very quickly, to perform this, what we're actually doing is inserting these thin film membranes into 3.5-inch sorbent tubes. These are then coupled to an adapter that allows the analyte to move from the membrane and in the sorbent tube up into a large bore needle trap device. This is done in a secondary, also portable high-volume desorption unit, shown in the bottom left-hand corner, and then inject this needle trap onto the portable GCMS instrument. And as you can see, by performing this coupling, we were really able to achieve good limits of quantitation using our portable instrument. We're actually seeing limits of quantitation that are relatively the same as our SPME experiments performed on a benchtop instrument, and even that US EPA liquid extraction approach, again on a benchtop instrument. Again, we're seeing this because we're combining a much more sensitive sampling approach with a less sensitive portable GCMS instrument. Furthermore, we were actually pleasantly surprised to see that we had good linear dynamic range even over 2 orders of magnitude, as shown below.
My name is Natalie Reyes Garces. I'm a postdoctoral fellow at University of Waterloo, and I'm going to describe some applications of SPME in bioanalysis. First of all, SPME has been successfully used for high-throughput analysis of urine. In the application that is presented in the slide, a C18 coating was employed for analysis of 110 different substances that were representative of various classes of doping compounds. The sample preparation process simply involved mixing urine with a 2 molar phosphate buffer to adjust the urine pH, as well as the addition of internal standard. Neither centrifugation nor adjustment of urine ionic strength was required. Then, by performing conditioning, extraction, wash in water, and desorption of the extracted analytes, it was possible to attain satisfactory figures of merit for the majority of the compounds. As shown, in terms of accuracy, most of the compounds exhibited less than 5% deviation from the normal concentration at 3 different concentration levels. Similarly, high analytical precision was attained by employing the Concept 96 automated workstation. It is worth emphasizing that by taking full advantage of the 96 samples that can be processed simultaneously, a total time of 1.7 minutes can be attained per sample. By following a similar approach, satisfactory analysis of prohibited drugs in plasma samples was also possible. In this study, 25 compounds were successfully analyzed in plasma by employing a coating made of polyacrylonitrile and HLB particles. The use of HLB demonstrated to provide improved extraction of polar drugs. In addition, satisfactory figures of merit in terms of accuracy and precision were obtained. This methodology proved to be highly convenient as minimum sample preparation was required. And it is also important to mention that SPME does not have any issues related to clogging of cartridges as it can happen in SPE. The use of biocompatible coatings has also facilitated the analysis of whole blood. Satisfactory results were obtained for a wide range of analytes when HLB pan immobilized on polybutylene terephthalate rods was used for the analysis of blood via direct immersion. For this particular work, the introduction of 3 washing steps instead of only 1 enabled remarkably clean extracts suitable for analysis via LCMS. As shown, absolute matrix effects ranging from 80 to 120% were found in urine, plasma, and whole blood for most of the target analytes. These findings support the capabilities of SPME in providing outstanding sample cleanup when dealing with highly complex matrices. Biocompatible SPME devices have been also employed for the analysis of fatty acids in biological fluids. It is worth mentioning that such non-polar analytes display high affinity for matrix components such as albumin, and therefore they have low free concentrations. In this study, it was possible to determine the plasma protein binding for 9 different fatty acids. In addition, the use of SPME permitted to determine fatty acid concentration changes in plasma samples of patients undergoing cardiac surgery with the use of a cardiopulmonary bypass. SPME has been also employed for metabolomics studies of clinical interest. As shown in the slide, there are several features that make of SPME a useful technique for such type of studies. The main feature, I would say, relies on the suitability of SPME to be used in vivo for in vivo sampling. By integrating sampling and sample preparation in a single step, metabolism quenching is facilitated and risks of alteration of the metabolome are certainly minimized. In addition, as the SPME coating enables balanced metabolite coverage, a broad range of metabolites can be concomitantly extracted. The biocompatibility of the coatings, their high selectivity for small compounds, and their flexibility as they can be easily mobilized on different support configurations are also important features of SPME when considering to use this technique for metabolomic studies. As it is presented in the slide, in an in vivo study where neurotransmitters were monitored in rat brains in vivo after fluoxetine administration, SPME and microdialysis provided comparable results. As microdialysis is the standard technique for in vivo brain analysis, the results obtained in such work endorse the applicability of SPME for such investigations. It is worth mentioning that SPME extras do not contain salts, which makes them highly compatible with MS or mass spectrometry systems. In this slide, it can be observed that in terms of metabolite coverage, SPME and microdialysis provide complementary information. SPME is able to facilitate the extraction of less polar metabolites, whereas microdialysis provides better coverage for more polar compounds. In a recent work that is being conducted in our lab, SPME is being used to investigate metabolic changes occurring in rat brains after deep brain stimulation. As SPME is a non-lethal technique, animals can be sampled more than once. Although such study is not published yet, it is worth mentioning that very interesting findings have been obtained. One of them, for instance, was the capability of observing changes in citrulline levels after deep brain stimulation. This finding could be an indicator of an increase of nitric oxide synthesis as a result of DBS. This work is the first untargeted study that is being conducted to investigate DBS mechanisms.
Hello everyone, my name is Barbara Boyko. I'm a former research associate in Professor Pawliszyn's group and currently I'm associate professor at Nicolaus Copernicus University in Poland. My research interest is focused on clinical and pharmaceutical applications of solid phase microextraction, and this will be the topic I will present to you here. In the previous section, my colleague described to you the features of bio-SPME. In this part, I would like to emphasize which of these features are particularly important in clinical analysis. In case of traditional sample preparation methods, solid and liquid matrices require significantly different protocols, such as centrifugation of biofluids to remove solid matrix components, or weighting and homogenization of tissue prior extraction with solvents. These, however, are not the procedures preferable in the clinical environment, especially for on-site analysis, as they decrease the overall throughput of the analysis. In SPME, which does not require any sample pretreatment mentioned before, the protocols are quite uniform and straightforward, consisting of preconditioning, insertion of the SPME device to the matrix, and then its removal followed by quick wash and desorption. As many bioapplications require high throughput and automation, the autosamplers which are shown in this slide and which were also described in the previous sections have a defined place in a clinical labs too. However, the unique feature of the method we are presenting to you, solid phase microextraction, is the possibility of conducting low invasive in vivo and in situ analysis of tissue. Sampling can be easily done on site, it does not require trained analytical chemists, and it can be done by medical personnel such as surgeon, Fibers can be sterilized by accepted methods, for example autoclaving, so there is no risk of infection, and the small size of the probe allows repeated sampling that we may obtain information at different time points, which might be of great importance, and I will show you this in the next slide. As it was already explained by my colleagues, SPME can be used for targeted and untargeted analysis. Therefore, the range of applications is very broad. For instance, we may first search for biomarkers of given cancer, and after validation of the selected compounds according to established regulations, the method can be used for targeted analysis. And here I would like to refer to the next part of the presentation about direct coupling of SPME to mass spectrometer, because the strategies described there can be successfully used for rapid diagnostics on site, including intraoperative analysis. Just to illustrate the usefulness of in situ tissue analysis with SPME, I would like to use an example of organ transplantation. Right now, the decision if the graft should be transplanted or not is made mainly based on visual inspection of the organ. In some countries like US, additionally a biopsy is taken in case of some organs. However, the quality of the graft changes, it declines during preservation because of the prolonged ischemia and oxidative stress. Therefore, a tool which would allow for monitoring of these changes over time from harvesting to transplantation would be appreciated. So SPME can be used for this purpose, first to identify biomarkers which would indicate the state of the graft, and then for the determination of those biomarkers on site. The proof of concept study was already reported on animal model for lung and liver grafts. As you can see on the left side of the slide, SPME fibers can be either assembled in the needle and they might be used or they might be used without any assembly to decrease the diameter of the device and so consequently the invasiveness of sampling. The two photos of lungs on the bottom left show the sampled area during in vivo sampling in triplicate and after fibers removal. It can be seen that there was no bleeding observed, and it confirms minimum invasiveness of the approach. Below the photos you may see the principal component analysis PCA plot with two clusters of data representing lungs subjected to two different methods of preservation: a normothermic ex vivo perfusion, this is the one on the left, and static cold storage, the one on the right. You may find more details in the original paper, but this example shows that we are able to extract a set of representative compounds which can be considered as potential biomarkers when studies involve appropriate bigger cohort. On the right of the slide there is again a PCA plot, but this time showing separation between the data obtained from plasma samples collected from the recipients of liver harvested from heart-beating donors, which means dead donors, and from living donors, so on the left and right side of the graph respectively. In case of heart-beating donor livers, it is expected that their performance might be affected by longer time of ischemia when compared to living donor organs. Therefore, this approach demonstrated that metabolomics can be used for post-transplant monitoring, because the separation is driven by the endogenous metabolites, which again can be used as biomarkers after required validation. Examples of the discriminant metabolites can be found in the original article. It was already mentioned in the previous section that SPME can be successfully used for drug analysis, and in the same study of post-transplant assessment of liver function, it was proposed that drugs routinely administered during transplantation protocol could be potentially used as markers of organ function. And indeed, it was confirmed that rocuronium, the drug which undergoes mainly hepatic clearance, was characterized by increased blood concentration in the patients who received liver from heart-beating donors, while tranexamic, which elimination is determined by kidney, did not show such a relation. The last example of the SPME fitting clinical analysis from bench to bedside is presented on the next slide, and the figure shows two approaches to cancer study. Again, sampling can be done in situ from the collected material, in this case resected brain tumor, or directly from the tumor in vivo. So as previously in the first step, the goal is to find metabolites specific to type of cancer or biomarker which would permit determination of cancer malignancy. And in fact, there are a number of cancers where histology, used to consider as a gold standard in cancer diagnostic, fails in terms of prognostic value, and genetic or metabolic markers are sought instead. So having an analytical tool which is suitable for fast intraoperative analysis would allow avoiding delays in decision-making process about patient treatment, and also the in vivo sampling could be of particular importance in cases where access to tumor is restricted and it cannot be removed completely, but SPME probe could be inserted in vivo to cancerous tissue to characterize the type of cancer and degree of malignancy. And now we switch to cell, microsomes and bacterial studies. So cell or bacteria culture studies with SPME are relatively common, but mainly in terms of analysis of volatile compounds from the headspace of the culturing system. For that purpose, one can use either manual SPME sampler or automated system like SPME GC autosampler. You can see the simple scheme of such manual approach on the bottom right of this figure. More recently, direct immersion mode of SPME was implemented to in vitro assays dedicated to cell, bacteria and related studies. These analyses can be done directly in the 96 well plates of various sizes thanks to the compatibility with the available SPME autosamplers. Also, the researcher may pick between the thin film format or regular SPME format. The first is rather preferable if single point extraction is performed because it may cause depletion of metabolites in the studied system, thus shifting its equilibrium. But on the other hand, it allows for significant increase of sensitivity compared to fiber. The fiber format is recommended for time course analysis when non-depletive extraction is required. The latter approach has great advantage over traditional assays which require stopping the metabolism and addition of dedicated dyes to the wells. Therefore, analysis in subsequent time points need to be performed on different samples, wells. And in SPME protocol, the sample in each well can be subjected to extraction number of times as long as no depletion of the target analyte is observed. So it should be emphasized here that it is not only cost-effective and time-efficient approach, but it also permits to avoid interwell variability and consequently to obtain more reproducible results. The last point which should be mentioned here is that all these in vitro assays may be targeted or they may consider untargeted studies such as profiling of metabolome or lipidome of the cells. So one may observe the changes of administered tested drug or active substance and monitor the response of the cultured cells simultaneously. On this slide you may find three examples of the SPME studies related to in vitro assays. So the first on the left presents the profiling of volatile metabolome of human-induced pluripotent stem cells along their in vitro differentiation. And as indicated in this panel, the authors were able to successfully identify compounds which were characteristic for the subsequent steps of the cell differentiation. The SPME GC data was also compared with the results obtained by sensor arrays, and for details please refer to the original paper. The middle panel shows the data from the paper focusing on optimization of the time course analysis of three compounds varying in physicochemical properties which were extracted from the low volume well plates, and the authors describe all factors which needs to be taken into account when performing, for example, cytotoxicity test or similar studies using SPME. And the last panel on the right refers to the research where SPME was used for untargeted screening of unknown phase 1 metabolites of combrettastatin A4 using rat liver microsomes. And here the time course analysis enabled to observe trends between decreasing level of parent drug and increasing concentration of its metabolites. The metabolites were identified based on the accurate mass and fragmentation pattern and compared with the in silico prediction of metabolism pathways. So as you can see, the flexibility of SPME allows to utilize the method in various areas of medicine, particularly direct clinical practice, but also in preclinical research using high throughput in vitro assays. What is important, the methods do not require modification of existing protocols, so they could be easily adopted in the labs and ICU or OR units.
Hello everyone, my name is Herman Gomez and I'm going to be talking about solid phase microextraction directly coupled to mass spectrometry. In recent years, there have been a growing need for technologies that allow us monitoring pesticides in food matrices, pharmaceuticals in environmental waters, and drugs and or their metabolites in health-related samples such as blood, plasma, and urine. Now, these technologies need to be rapid, they must be simple so they can be easily to operate, they should provide data ideally close to real time, they must be deployable, and hopefully they can perform high throughput analysis. But that's not the only things. These technologies should also be capable of performing clean analysis, and in terms of clean, that they will not contaminate our instrumentation, particularly the mass spectrometers. They must be reliable so we can trust the data we're generating. They should be sensitive to the compounds we're interested in, selective so we have less chances of false positive, and hopefully we can go after multiple targets. The problem with the current technologies and most of the things you have seen in this review is that the direct interface with mass spectrometry is done via chromatographic step, and as a result that dramatically increases the total analysis time. In recent years there have been a big growth in technologies that are focused on the direct interface of solid phase microextraction to mass spectrometry, and some of these technologies we can highlight the in-tube SPME, the solid phase microextraction coupled to mass spectrometry either via nanoelectrospray ionization, open pore probe, the microfluidic open interface, or the dielectric barrier desorption ionization. We also have the solid phase microextraction transmission mode or SPME mesh, that coupled to direct analysis in real time, and the coated blade spray. So when we move to this slide number 2, we can see that there is a clear difference between the classical ambient ionization technologies or direct sample to MS technologies and the SPME MS technologies. And essentially it is that in the case of SPME, rather than just placing the sample in front of the instrument for analysis, what we do is that we perform a rapid enrichment and exclusion of macromolecule salts or lipids. By doing this, that will allow us reaching lower limits of detection and much better limits of quantitation. After the extraction of the analytes, the coatings are exposed to a rapid cleaning in water, and then they are brought in front of the mass spectrometer for analysis. So this slide portrays the direct interface via a direct analysis in real time source. So then the heated gas touches the surface of the mesh, and then the analytes are thermally desorbed into the instrument. The advantage of SPME versus the conventional ambient mass spectrometry and direct sample interfaces is that they will allow us introducing a much cleaner matrix into the instrument. But there are certain differences. Now that we are in slide number 3, we will see some of them. So the classical analysis time, as can we see in the figure B, it's measured in the order of minutes. So we can have a chromatographic separation, and then the analytes will arrive at different times to the mass spectrometer. What we're trying to do in order to dramatically decrease the analysis time by SPME MS is to just insert all these analytes into the instrument at the same time, so we can have analysis in the second, in the order of a few seconds. So the figure number 8 portrays what we typically would see by technologies such as dielectric barrier desorption ionization, DBDI, the open pore probe, OPP, or the microfluidic open interface, MOI. Now, figure number C portrays what we'll see when we are using technologies that allow us having a transient signal, such as the coated blade spray and the nanoelectrospray ionization. But there are advantages and disadvantages to both of these approaches. So now, as can be seen in figure D, one of the advantages of the technologies such as OPP and DBDI is that all the analyte will be introduced into the mass spectrometer at once, versus in technologies like blade spray and nanospray, the analyte can be introduced over a long period of time. So the advantage of the first ones is that we will have a better signal to noise ratio, will allow us to have a better limits of quantitation, while the second technology will allow us to perform much more MS events, so we can perform like a larger number of analytes can be quantified.
I